Recent analytical developments from the Massachusetts Institute of Technology have introduced a significant shift in the discourse surrounding domestic biosurveillance, specifically regarding the prevalence of hantavirus. Mathematical models produced by researchers suggest a predictive framework in which hantavirus, once considered a primarily regional concern associated with specific ecological niches in the American Southwest, could potentially manifest in environmental reservoirs across all 50 states. While the terminology used in some initial reporting characterizes this expansion as terrifying, public health officials and virologists urge a more measured, data-driven approach to understanding what these models actually signify for human populations. To understand the gravity of these projections, one must first understand the fundamental nature of hantaviruses. They are a family of viruses spread mainly by rodents and can cause varied disease syndromes in people worldwide. In the Americas, the primary concern is hantavirus pulmonary syndrome, or HPS, a severe and sometimes fatal respiratory disease. Each hantavirus serotype has a specific rodent host species and is derived from a long-term evolutionary relationship with those hosts. The most well-known in North America is the sin nombre virus, carried by the deer mouse, though other reservoirs include the white-footed mouse, the rice rat, and the cotton rat. The framing of this new MIT study suggests that shifting climatic variables and migratory patterns of these rodent hosts could lead to a presence of the virus in areas previously considered low risk or dormant. This news comes at a time when global public health infrastructure is increasingly focused on predictive modeling to prevent zoonotic spillovers before they reach a critical mass. The significance for the viewer lies in the potential for increased exposure in suburban and urban environments where rodent-human interactions were previously thought to be minimal. However, it is essential to distinguish between the presence of a virus in a rodent population and an active outbreak in the human population. Historically, hantavirus infections in humans are relatively rare, but the high mortality rate associated with the pulmonary syndrome, estimated at approximately 38%, necessitates rigorous monitoring. As these mathematical models suggest a widening geography for the virus, the emphasis shifts from reactive clinical treatment to proactive environmental surveillance. This report will examine the mechanics of these predictions, the current state of laboratory readiness, and the specific guidance provided by health agencies to mitigate risk as the ecological footprint of these reservoirs continues to evolve. The core of the recent reporting centers on the complexity of mathematical modeling used by institutions like MIT to track viral movement through ecosystems. These models do not operate in a vacuum. They integrate massive datasets including precipitation levels, vegetation density, and rodent population dynamics. In the field of epidemiology, a trophic cascade often explains sudden spikes in hantavirus presence. For example, increased rainfall can lead to an abundance of seeds and fruit, which in turn causes a boom in the rodent population. As these populations expand, they may move into new territories or closer to human dwellings, increasing the statistical likelihood of viral transmission. While the headline figures from the MIT model suggest a national spread across all 50 states, it is crucial to analyze how agencies like the Centers for Disease Control and Prevention and state-level health departments verify such claims. Currently, surveillance involves the trapping and testing of rodents in various habitats to determine the viral prevalence rate within a specific community. Lab work is highly specialized because hantaviruses are handled under biosafety level 3 conditions. Testing is often centralized in major state or federal facilities. Common diagnostic methods include enzyme-linked immunosorbent assays, or ELISA, to detect antibodies and polymerase chain reaction, or PCR to identify the viral RNA itself. The discrepancy between the terrifying spread predicted by computers and the actual confirmed human cases often stems from the difficulty of transmission. Unlike many respiratory viruses, hantavirus is not known to spread from person to person in North America. Infection typically occurs when a person breathes in air contaminated with the virus through aerosolization. This happens when rodent urine, droppings, or nesting materials are disturbed, sending microscopic particles into the air. Therefore, even if the virus is present in all 50 states as the model suggests, the actual risk to the public remains dependent on specific behaviors and environmental exposures. Public health officials emphasize that while the model is a valuable tool for preparedness, it does not imply a sudden surge in hospitalizations. Instead, it serves as a signal for clinicians nationwide, not just those in the West, to include hantavirus in their differential diagnoses when a patient presents with unexplained severe respiratory distress and a history of potential rodent exposure. The labs remain the final word in this surveillance chain, moving beyond theoretical models to provide the empirical evidence required to trigger official health alerts or travel advisories, though no such restrictions are currently indicated by the source material. Given the expanding geographical footprints suggested by modern modeling, public health guidance has become more uniform and urgent. Prevention remains the primary defense against hantavirus, as there is currently no specific vaccine or cure for the infection. Health agencies advise a clean and clear approach to any area where rodents may have been present. This includes seasonal cabins, sheds, barns, and even parked vehicles. The standard protocol for cleaning involves a mixture of water and household bleach, which effectively deactivates the virus. Crucially, officials warn against sweeping or vacuuming dry droppings, as this is the most common way for the virus to become aerosolized and inhaled. Instead, the area should be thoroughly soaked with disinfectant before being wiped down with paper towels. For those in high-risk professions or those engaging in heavy cleaning of infested areas, the use of personal protective equipment, including gloves and high-efficiency particulate air respirators, is recommended. Surveillance continues to be a collaborative effort between ecologists and virologists, who monitor sentinel populations of mice to catch early signs of viral spikes. Despite the advancements in predictive technology, significant gaps in knowledge remain. Scientists are still investigating why certain individuals develop the severe pulmonary form of the disease, while others might have a more muted response, and the long-term impact of climate change on rodent migration remains a subject of intense study. The uncertainty inherent in mathematical models means that while the all 50 states scenario is a possibility for which we must prepare, it is not a guaranteed outcome. Public health reporting serves to bridge the gap between these high-level scientific predictions and the everyday reality of the citizenry. By maintaining a calm and informed perspective, individuals can take the necessary steps to rodent-proof their homes and recognize the symptoms of infection early. These symptoms typically begin with fatigue, fever, and muscle aches, which can be easily mistaken for the flu, making a history of potential exposure a vital piece of information for any medical consultation. As we continue to monitor the data from MIT and subsequent reports from the field, the focus remains on education, early detection, and sensible environmental management. This coverage will continue as more data becomes available from official monitoring stations. For more updates on global health reporting and scientific developments, consider subscribing to this channel. The core of the recent reporting centers on the complexity of mathematical modeling used by institutions like the Massachusetts Institute of Technology to track viral movement through domestic ecosystems. These models do not operate in a vacuum. They integrate massive environmental datasets including precipitation levels, vegetation density, and rodent population dynamics to forecast potential zoonotic shifts. According to the projections, the prediction of a hantavirus presence across all 50 states represents a significant departure from the traditionally understood geographic range of the virus, which was historically centered in the American Southwest. To understand why this model is viewed as a critical development by the scientific community, one must look at the infrastructure of domestic public health surveillance and the specific laboratory protocols required to identify these pathogens. Unlike more common seasonal viruses, hantaviruses are categorized as special pathogens, requiring high containment facilities, specifically biosafety Level 3 labs, for any work involving the live virus or the processing of highly concentrated samples. The diagnostic pipeline for hantavirus is famously rigorous. When a potential case reaches a hospital or clinical setting, the initial suspicion often arises from a patient's history of exposure to rodents combined with specific clinical markers, such as a rapid drop in platelet count and the presence of immunoblasts, which are atypical lymphocytes, in the blood. Once a sample is sent to a state laboratory or the Centers for Disease Control and Prevention, technicians utilize two primary methods for verification: enzyme-linked immunosorbent assays, or ELISA, and reverse transcription polymerase chain reaction, known as RT-PCR. The ELISA test is designed to detect specific antibodies, namely IgM and IgG, which represent the body's immune response to the viral threat. IgM antibodies generally indicate a recent or acute infection, while IgG can persist for years after exposure. The RT-PCR method, conversely, looks for the actual genetic material of the virus in blood or tissue samples, allowing for a definitive identification of the specific viral strain. The MIT model's assertion that the virus could be present in all 50 states necessitates a fundamental shift in how clinicians nationwide evaluate severe respiratory illness. Historically, hantavirus pulmonary syndrome has been most famously associated with the deer mouse, or Paromyscus maniculatus, particularly in the Four Corners region. However, other known strains like the New York virus, carried by the white-footed mouse, and the Black Creek Canal virus, carried by the cotton rat, have already demonstrated that the reservoir is not limited to a single species or region. The mathematical modeling likely accounts for these various reservoirs and the ways in which their habitats are shifting due to land use changes and climatic variability. In laboratory settings, researchers also focus on the viral load found in these rodent populations. Not every rodent in an area carries the virus, and the amount of virus shed into the environment can vary based on the animal's age, health, and the season. This variability is a key component of what scientists call a trophic cascade. This phenomenon occurs when environmental changes, such as an unusually wet winter, lead to an abundance of food sources like seeds and nuts. This leads to a boom in the rodent population, which in turn increases the density of the viral reservoir near human dwellings. While initial reporting describes the MIT model as terrifying, it is essential to contrast this with the verified mechanics of transmission. Hantavirus is not an airborne virus in the same category as influenza. It does not travel long distances through the air or survive indefinitely on most surfaces. It requires the direct aerosolization of fresh rodent excreta, typically in enclosed, poorly ventilated spaces. Therefore, a predicted presence in all 50 states does not automatically correlate to a high volume of human cases, provided that effective public health communication and environmental controls are maintained. The core development here is the transition from reactive medicine to a more predictive, data-heavy surveillance model that utilizes the computing power of major research institutions to stay ahead of ecological shifts before they manifest in clinical settings. In response to the expanded risk profiles suggested by the MIT mathematical models, public health agencies are reinforcing standardized protocols for prevention and environmental management. Since there is currently no vaccine, antiviral treatment, or specific cure for hantavirus pulmonary syndrome, the medical community relies entirely on supportive care and rigorous primary prevention. The most critical practical takeaway for the public involves the management of rodent-infested spaces. Health officials emphasize that the virus is primarily transmitted through the inhalation of aerosolized particles from rodent excreta. Consequently, the standard recommendation for cleaning any area where rodents have been present is a strict wet cleaning method. This involves saturating the affected area with a disinfectant, ideally a solution consisting of 1 part household bleach to 9 parts water, and allowing it to soak for at least 5 minutes before removal. This process is designed to deactivate the virus and, more importantly, to prevent contaminated dust from becoming airborne. Public health guidance explicitly warns against the use of vacuums or brooms in these settings, as these tools effectively launch viral particles into the breathing zone of the individual. For those tasked with cleaning heavily infested areas, the use of personal protective equipment is non-negotiable. This includes non-porous gloves, eye protection, and high-efficiency particulate air, or HEPA, filtered respirators. Beyond immediate cleaning, long-term prevention focuses on exclusion and habitat modification. This entails sealing any entry points in a structure larger than 1/4 of an inch, roughly the width of a pencil, using materials that rodents cannot easily chew through, such as steel wool or metal flashing. From a surveillance standpoint, the MIT findings suggest that health departments in states previously considered low risk must now establish more robust ecological monitoring programs. These programs typically involve the trapping of sentinel rodent populations to assess the seroprevalence of the virus within a given area. This data provides an early warning system that allows officials to issue localized alerts before human cases emerge. However, there are significant limits to current scientific knowledge that must be acknowledged. While mathematical models can predict the presence of a virus, they cannot yet account for the complex interplay of human behavior and individual immune responses that lead to a spillover event. It remains unknown why some individuals exposed to the virus develop the fatal pulmonary syndrome, while others may experience milder symptoms or avoid infection entirely. Furthermore, the terrifying spread predicted by these models remains probabilistic rather than a certainty. The ethical responsibility of public health reporting in this context is to balance necessary awareness with a rejection of alarmism. The presence of a viral reservoir in all 50 states does not equate to an inevitable pandemic, given the virus's inability to spread between humans in North American strains. Instead, it serves as a call for heightened clinical vigilance and improved domestic sanitation. By understanding the mechanics of aerosolization and the simple efficacy of household disinfectants, the public can mitigate much of the risk highlighted by these advanced computational models. As the scientific community continues to refine these predictions with real-world field data, the focus remains on transparent communication and the maintenance of a calm, informed readiness. For more updates on the intersection of advanced modeling and global public health, consider subscribing to this channel.